Okay, so welcome back everyone. I've just got a couple of reminders to begin with. So one is that the midterm test is in 2 weeks' time, in week 7. So that will be held in the usual lecture slot for that week here in LT11. We mentioned earlier in the course that it's going to be made up of multiple response questions, and there's some more information about the format of the test in the tutorial that's running this week for those who haven't had that tutorial yet. Yesterday I posted a practice quiz on Canvas, and this has quite similar types of questions as what will be on the midterm test, and so I'll encourage you to do that as part of your revision. I also posted a feedback survey on Canvas, which will be up until the end of recess week. And so if you have any feedback on the lectures, the tutorials, any other aspect of the course, then please let me know. And then I'll have that in mind when I'm finalising the content for the second half of the semester. There's been a small update to the schedule of tutorials, so just switching 2 of them around. So in week 7 and 8, there will be a tutorial which is a discussion of a paper on anxiety disorders from an evolutionary perspective. And then the tutorial in week 9 and 10 will be including feedback on the midterm test. Okay, so this week we're continuing with the topic of vision and focusing a bit more on the role of the brain in vision. To introduce the topic, I'm going to tell you a little bit about this quite influential clinical case, influential in understanding of visual neuroscience. It's a particular woman who's known in the literature as patient LM. And she suffered a stroke when she was in her 40s. And following her stroke, she had this really peculiar symptom, which is that she was then unable to see the movement of objects around her. So she describes her experience of the world. Her visual experience has been like a series of kind of snapshots or still images where objects might kind of change position from moment to moment, but she wouldn't have any sense of the kind of intervening movement that led to that change in the position of the object. This is called achinotopsia, this inability to see motion, and it's been described in just a very small number of clinical cases following damage to the cortex. And there's a video here that tries to give a bit of a picture of what her experience is like.
This woman suffered a stroke 14 years ago. Now her view of the world is dramatically skewed. She cannot detect how full the glass gets. She sees the water, but she doesn't see the water rising. She can no longer detect motion. The stroke interrupted the neural pathways that process motion, the where of the visual scene. Hers is the only reported case of its kind. The world her brain sees for her is terribly disjointed. Simply watching a car pass by can be a frightening experience. She sees in freeze frame. Her physical world is made up of disjointed still images, like the pictures on a film reel, except there are large gaps between each of them. A mere stroll down the street can be terrifying, for there are so many moving objects to understand in those flashing still frames. And many of those moving objects could hurt her. When both she and the object she is looking at are moving, things get even worse. It probably looks something like this. When just one of the many parts of the brain's visual system is impaired, our view can be dramatically altered.
Okay, so there have been many research studies that she's participated in, and which have tended to confirm that she has this specific inability to see movement. And these are some quotes that help to sort of further illustrate what this is like for her. So she describes how people, dogs and cars appear restless, are suddenly here and there, and then disappear in between. Fluids appear frozen like a glacier, which causes difficulty with pouring tea or coffee into a cup. Filling a glass with water becomes impossible. She finds it very irritating to meet people and talk to them because she can't respond in time to their handshake, and because she finds their moving hand disturbing. She has to avoid watching their facial expressions. Their lips seem to jump rapidly up and down, and this kind of distracts her from what they're saying. But in contrast, when people, faces, objects and cars are stationary, she can recognize them immediately and accurately. So the perception of color doesn't change, and she reports having no difficulty with perceiving the position of objects or judging how far away they are and the distances between them. And so this kind of case illustrates something profound about our perception, about the modularity of our perception, which is that, or to illustrate, when we see something around us, like we see a kind of red rolling ball, we experience that visually as kind of a single event. But there are different characteristics to this experience that we can distinguish. Like the color of the ball is red, its shape is round, it's moving towards the left, it's a ball as opposed to some other type of object, it's a particular distance from us, it's a particular size, and so on. And these different features of our experience are produced partly by different mechanisms in our brain, so different networks of neurons that are specialized to take the signals coming into the brain from the retina and extract these different types of information. And so that's what makes it possible for somebody like LM, who has suffered damage to a particular part of her cortex, to lose the ability to see movement while still being able to see all the other properties of objects around her. So in this lecture, we're going to talk about how signals are sent from the retina into our brain, the pathway they take through our brain. I'm going to talk about the feature selectivity of neurons in our visual system, so meaning the property of neurons in our brain that they tend to respond to specific types of features of what we're seeing. We'll start with early stages of visual processing in the cortex, where there are neurons that respond to very simple features of our vision. And then we'll talk about higher level visual processing, meaning the later stages of cortical processing of visual information, where more complex, kind of meaningful information is extracted, so underlying our ability to recognize other people, for example. So by the end of the lecture, you should be able to describe the pathway that visual information takes from our retina into our brain, explain why cells in the visual system respond selectively to specific visual features and how this has been investigated, and contrast the function of the dorsal and ventral pathways of vision, which I'll describe later in the lecture. So last week we talked about the retina in some detail. We saw how light entering our eye collides with the photoreceptors, the rod and cone cells that line the back surface of our eye. That generates electrical signals that are passed on to bipolar cells in the retina and then passed on to the ganglion cells in yellow here. And the ganglion cells have axons that extend from the retina through the optic nerve into the brain. And so the initial input that our brain is receiving about the outside world comes in the form of a pattern of action potentials that are travelling down the optic nerve. The optic nerve projects from the retina in our eye to a part of the thalamus. And so in the lecture on anatomy, we talked about how the thalamus is this subcortical structure deep in the centre of our brain that acts as a kind of sensory relay station. So it receives signals from our retina. It also receives signals from our ears and our skin, for example. And then there are these tracks that extend from the thalamus to different parts of the cortex that carry that sensory information to different parts of the cortex. In the case of vision, the optic nerve projects to a particular part of the thalamus called the lateral geniculate nucleus, or LGN. And from there are axons that extend into a part of our occipital lobe, in particular a region called the primary visual cortex, or V1, which is an area at the very back of our occipital lobe, at the back of our head. Neurons in the primary visual cortex are receiving this information that's been relayed from our eyes, and then that information is relayed further into the brain, so into the parietal cortex at the top of our head and into the temporal cortex on the sides of our head. And so our visual system doesn't just include our eyes, of course, it's extending throughout much of our brain as well. What we'll see in this lecture is that cells at different stages of this visual pathway are responding to different features of what we're seeing. So in the retina and in the LGN, the lateral geniculate nucleus, we have cells that respond to very simple properties of the light that's falling on the retina. So basically to spots of light. Once we get to the primary visual cortex, we find cells that are responding to slightly more complex features of what we're seeing, which are things like edges and the orientation of those edges. And then later in the cortex, we begin to find cells that respond to more complex visual patterns, like a particular person's face or a complex kind of meaningful pattern of movement, like what you see here. And so you can think of the visual system as having this hierarchical organization where earlier stages of visual processing are extracting simpler features of what we're seeing, and then later stages build on that by extracting more complex patterns. So the key points here are that signals from the retina, from the ganglion cells, are relayed to the primary visual cortex in our occipital lobe via the thalamus. And then there are these neural pathways that extend from the primary visual cortex further across the cortex into the temporal and parietal lobes that enable more complex processing of visual patterns. So in the second lecture of the course, when we talked about the action potential, I briefly talked about these studies that were done by Hubel and Wiesel, where they recorded the electrical activity, the action potentials that were occurring in individual cells in the primary visual cortex and other parts of the brain in cats. So that they were recording the activity of one cell at a time by inserting a microelectrode into the cat's cortex. And then they showed the cat different images on the screen and varied the content of those images to try to find whether there was some type of stimulus that would produce activity in that cell that they're recording from. And so these experiments were quite important for our understanding of how what the role of our brain is in vision. And so I'm going to go through these in a little bit more detail now. To start with, a concept that's useful to define is the receptive field of a neuron. And so this refers to the particular part of our visual field, what we see in front of us right now, that the neuron is responsible for, or which it responds to. So to illustrate, if we consider a particular photoreceptor in our retina, so a rod cell, for example, that's located sort of closer to the top of the retina, because of the geometry of the eye, that photoreceptor will only be stimulated by light that's coming through the eye at a specific angle. So in the case of this photoreceptor located more towards the top of the eye, it will only be stimulated by light that's coming through, entering our eye sort of from the bottom. And so correspondingly, there's a particular part of our environment or a particular part of our visual field where if light is generated there, then this photoreceptor can detect that light. And so that's the receptive field of that photoreceptor. In the previous lecture, when we were talking about the retina, we saw how ganglion cells are often receiving signals from a number of different photoreceptors at the same time. And this is particularly the case in the periphery of our retina, where there can be a number of different receptors all located sort of in a patch together on the retina that will all be sending excitatory signals to one particular ganglion cell. And so we can then define the receptive field of that ganglion cell as opposed to one of the receptors. And because it's receiving signals from a patch of our retina, there will correspondingly be kind of a slightly larger patch of our visual field where light can come from to stimulate this ganglion cell. So the receptive fields of ganglion cells are also illustrated in this video. And so this is from one of Huber and Wiesel's experiments where, again, they're inserting an electrode, in this case, to come in contact with cells in the thalamus that are receiving signals from the ganglion cells of the retina. And what you're seeing in the video is effectively what the cat is seeing during the experiment. And this crackling sound that you hear that you will hear in the background represents the electrical activity of the cell that they're recording from in real time. Okay, so you can see here that this ganglion cell or cell in the LGN is responding to something very simple. It's just responding to a spot of light that's located in a particular part of the cat's visual field. So when they move the light away from that particular part that's circled, the cell stops responding, and this defines the receptive field of that particular cell that they're recording from. It turns out that the ganglion cells in our retina have something called a center surround receptive field, which is illustrated here. And this consists of a central part of the receptive field that is excitatory and a surrounding kind of donut-shaped region that is often inhibitory. So if we have a spot of light falling on our retina that is in just the right location of the retina and is just the right size to stimulate that excitatory central part of the receptive field, we'll get a strong response in the ganglion cell, so represented here as a high rate of action potentials over time. But if we increase the size of that spot of light, and it begins to stimulate the surrounding inhibitory region of the receptive field, then that leads to a reduction in the response of the ganglion cell. And so here this neuron, because of the presence of this inhibitory component of its receptive field, is responding best to a dot of light that has a particular size and also needs to be in a particular location of the retina. And we can understand why this occurs with reference to the wiring between neurons. So in particular, this figure here is kind of a simplified wiring diagram where it's meant to represent the photoreceptors, in this case rod cells at the top. So this is a group of rod cells all located next to each other on the retina that are all sending signals to this same ganglion cell. And there would also be bipolar cells in between the rod cells and the ganglion cell, but they're just omitted here for simplicity. But the idea here is that this central excitatory part of the receptive field corresponds to a set of rod cells that are all sending excitatory signals to this same ganglion cell. Whereas so if we have a spot of light falling just right on our retina to stimulate only those particular rod cells that are sending excitatory signals, we have this spatial summation of excitatory signals onto this ganglion cell that leads to a strong response in the ganglion cell. Whereas the photoreceptors that are located further away from the center here are forming inhibitory synapses with the ganglion cell. And so when light falls on that inhibitory surround of the receptive field, what's happening is it's stimulating those photoreceptors that are then inhibiting activity of the ganglion cell. And so in that case, the ganglion cell is receiving a mixture of both excitatory and inhibitory signals that overall leads to a lower response than in the previous situation. Okay, so the idea here is that we can understand the behavior of this ganglion cell in terms of its tendency to respond to a dot of light of a particular size and location in terms of the particular signals that it's receiving from photoreceptors. When Hubel and Wiesel then began to record from cells in the cortex, what they found is that they started with the primary visual cortex, so area V1, located in the occipital lobe. And what they found there is not cells that respond to dots of light, like in the retina, but rather cells that respond to slightly more complex features, and that's illustrated in this video. Okay, so they called these types of cells found in V1 simple cells. And what you can see in that video is that these cells are responding when light is in a particular part of the cat's visual field, highlighted here. But they're also responding to a slightly different type of feature, which is the presence of a bar of light or an edge that is at a particular orientation. And so you saw at the end there that as they kind of sweep the bar of light through that area at a different orientation, the cell doesn't respond to the same extent. So that idea is illustrated here as well, where In this particular example here, they're recording from a cell where when they present a horizontal bar of light within the receptive field of the cell, they don't get a response. But when they rotate it to vertical, then the cell starts responding rapidly, producing action potentials. And we can understand the behavior of this type of cell, again, with reference to its receptive field. And in particular, it can be understood in terms of a similar kind of center surround receptive field, but where the central excitatory part of the receptive field is kind of bar-shaped and at a particular orientation. And then it's surrounded by these bar-shaped inhibitory parts of the receptive field. And the consequence of that is that when there's an edge or a bar of light that's shining on our retina, if it's at just the right location and just the right orientation, then it will only stimulate that central excitatory part of the receptive field. So then we get a maximal response from the simple cell. But if the bar of light is oriented differently, parts of the light will begin to stimulate the inhibitory components of the receptive field, and as a result, we get a reduced response in the simple cell. And so this is illustrating the receptive field of a simple cell that is tuned to respond to vertical edges, but when they were recording from cells in the cortex, they found other cells that responded best to horizontal edges or to diagonal edges and so on. And we can understand why this occurs again in terms of the wiring of neurons and the kind of inputs that these simple cells are receiving. And so we talked just now about how ganglion cells in our retina are good at detecting dots of light of a particular size based off the pattern of excitatory and inhibitory connections or signals that they're receiving from the photoreceptors. And so if we want to detect a slightly more complex feature, like a bar of light with a particular orientation that's falling on our retina, we can do that by combining the responses of particular ganglion cells. So here what we're going to imagine is that we're looking at like a bird's eye view of our retina here. We have this vertical edge or bar of light shining on a particular part of our retina. And across the retina, there are ganglion cells with different receptive fields. And so 3 of those are illustrated here. And so these ganglion cells with the center surround receptive fields, if the bar of light is just at the right orientation to stimulate the center of those 3 ganglion cells, it will cause a response across those 3 ganglion cells. So we can think of this in terms of a wiring diagram as well. So here we have our 3 ganglion cells corresponding to these 3 center surround receptive fields. This is in the retina. And then in the cortex, we have a simple cell that is integrating together the responses of those 3 ganglion cells. It's receiving excitatory signals from each of those ganglion cells. And so when the bar of light falling on the retina is in just the right location to stimulate those 3 ganglion cells together, there'll be this spatial summation of excitatory signals that will lead to a maximal response from this particular simple cell in our cortex. Across the retina, we have lots of ganglion cells across the whole kind of breadth of the retina, each of which is attached to or receiving signals from a particular collection of nearby photoreceptors. And so here we're illustrating one particular simple cell in the cortex that's receiving signals from these 3 ganglion cells, but there'll be other simple cells that might be receiving connections from the 3 ganglion cells at the bottom here, for example. And in that case, that particular simple cell will respond to a horizontal edge falling on our retina rather than a vertical edge. Okay, so it's a very kind of simple example, but the idea here is that the behavior of cells in our visual system in detecting particular patterns of light, so here a vertical edge as opposed to a horizontal edge, for example, can be explained in terms of the spatial summation of signals that those cells are receiving from cells earlier in the visual system that are detecting simpler features. Okay, and so earlier in the course we've learnt about things like the spatial summation of signals and how that affects the behavior of a neuron, and the things like excitatory and inhibitory synapses. And here we're applying that to understand how neurons in the brain can come to represent information or patterns that are present in the light falling on our retina. In area V1 of our cortex, there are many of these simple cells, and these different simple cells will have different spatial receptive fields. They'll be receiving input from different parts of our retina, and they also, as I've noted, will prefer different orientations. So some will respond to vertical edges, others to horizontal edges, and so on. When we're looking at an object like this tree trunk, different parts of the object will fall into the receptive field of different neurons in our visual system. So the circles A, B, and C here are meant to be representing three different receptive fields. And so in our primary visual cortex, there will be neurons that are receiving signals that from those different receptive fields. And then amongst those neurons that are receiving signals from this particular part of the object, some of them will respond specifically to vertical edges, and so will be becoming active as we look at this object. And so the overall shape of the object is represented in the pattern of activity that it's causing across this large group of simple cells that are each interested only in a small part of the object and only interested in the presence of very simple features like a vertical edge or a horizontal edge. If we expand that to our entire visual field, or like a complex image like this, what these yellow ellipses are meant to represent are different receptive fields. So receptive fields for different neurons. And so the entirety of our visual field is sort of captured in the individual receptive fields of a large number of neurons. And again, each of those neurons only cares about a small part of our visual field and only cares about simple kind of information that's present there. But all that complex visual information that you see in front of you right now or in an image like this is initially being captured in terms of the response that it's causing across this large number of simple cells in our primary visual cortex. So edges are kind of very simple features and maybe not particularly interesting, but you may be able to imagine how if our brain is capturing this information about the orientation of all the edges in our visual field, it can then use that to detect more complex, kind of meaningful information as well. And so if you consider like the form of a person's face, for example, it can be captured in terms of a particular pattern of edges, like in this case, horizontal lines representing the eyes, more sort of vertical lines produced by the contour of the nose, and so on. And we've seen how our visual system is able to integrate responses from ganglion cells that detect just little spots of light to produce neurons that are able to detect slightly more complex features like edges. And similarly, our visual system is able to integrate together the responses of different simple cells that are detecting the presence of different edges in our visual field to produce neurons that can detect more complex features like the kind of general pattern of a person's face. And we'll talk a bit more about object recognition in the second half of the lecture. There's also been a bit of work looking at how the selectivity of cells in our visual system develops during our life. One well-known study by Blakemore and Cooper studied this in cats, and again looking at these simple cells in the primary visual cortex. What they did is they reared these cats from very early in life, for something like the first 6 weeks of their life, in an unusual visual environment. And so these kittens were kept in either sort of a dark area or in this kind of enclosure that was designed so that it had primarily just vertical edges. Okay, so the kitten wore a kind of cone to prevent it from seeing its own body, and so it's spending most of its time looking at kind of vertical edges rather than horizontal edges. That's the idea. After 6 weeks or so, they released these kittens and found that they were, much like other kittens. They played with each other and with people and so on, but they seemed to display some differences in their behaviour. So in their paper, they describe playing with the kittens with kind of like a rod-like toy, and the kitten would respond to the toy when it was waved sort of vertically, but not when it was waved horizontally. So giving some indication that it's responding to vertical versus horizontal edges differently. And then they also recorded activity in area V1, similar to the studies by Hubel and Wiesel. And what they found in V1 is in these cats reared in an environment with primarily vertical edges, they were able to find lots of simple cells in V1 that would respond to vertical edges, but they couldn't find simple cells in V1 that would respond when the cat was shown horizontal edges. And similarly, for cats reared in an environment with primarily horizontal rather than vertical edges, they found the opposite pattern. And so, suggesting that in these cats that early in life haven't been exposed much to a particular type of visual information, that they aren't developing cells in their visual system that are able to detect that information. And so it suggests that in us as well, that we're not just born with a visual system that's able to do everything that we're used to detect all the kind of information that we see in front of us now, but rather that ability of our visual system develops very early in life based off the kind of visual information that we're exposed to. Okay, so in the lecture so far, I've talked about how cells in our visual system respond to light, and due to the way that they receive signals from earlier stages of the visual system, they can detect features of what we're seeing, like a vertical edge, for example. And I think this is really important in explaining how information about the outside world is able to actually sort of end up in our nervous system, end up in our brain. And the assumption is that information or that behavior of the neurons in responding to something like a vertical edge also underlies or produces in some way actual conscious experience of seeing that vertical edge. But it's a deep and enduring problem in neuroscience and psychology and philosophy, understanding the relationship between biology and our conscious experiences. So the activity of our neurons in our brain and our actual experience of consciously seeing something in front of us, for example. And so we saw earlier in the course that the activity of neurons basically comes down to things like simple chemical events, like the movement of ions across the membrane of a cell and the release of neurotransmitters at the synapse. And we can show that empirically, that those kind of events are closely correlated with conscious experiences, that particular simple cells become active anytime we see a vertical edge, for example. But it's not obvious why those things should be correlated. Like, why couldn't our nervous system respond to light and extract that information regarding the presence of a particular feature, but without generating any actual conscious experience of seeing something. And so there's many theories about the relationship between the brain and the mind, it's also become increasingly trendy in the last decade or so, scientific research that tries to address this question. But currently we don't have any theory of the relationship between activity of the brain and consciousness that is actually convincing enough or supported by the evidence enough to become widely accepted. And so if you're interested in these kind of more philosophical questions, there's a part of the textbook that describes some of the theories of consciousness. There's also a 4,000 level seminar run by one of my colleagues, Camillo, that focuses on theories of consciousness. And so you might be interested in checking that out. Okay, so the key points here are that these cells very early in our visual system, like the ganglion cells in our retina, respond to very simple visual features like the presence of dots of light falling on a particular part of our retina. Once we get to the cortex, starting with area V1, we find cells that respond to other basic features like the presence of edges and the orientation of edges in particular locations of our visual field. And it's the wiring between neurons, the pattern of excitatory and inhibitory signals that they're receiving from earlier stages of the visual system that makes them respond to particular patterns that are present in the light falling on our retina. Okay, so we can have a break in a moment, but first let's do a poll everywhere question. So the question here is to imagine a visual system in which cells in V1 receive no inhibitory signals from the retinal ganglion cells that have been relayed via the thalamus. Instead, they only receive excitatory signals. And so which of the following outcomes do you think is most likely? Do you think there'll be improved detection of the orientation of edges due to the greater excitation of V1 cells? Do you think there'll be a loss of orientation selectivity in V1, meaning that the simple cells are not responding to specific orientations, but instead there'll be an elevated response to diffuse illumination of the retina? Will there be difficulty recognizing complex visual patterns like faces, or do you think more than one of the above is correct? All right, so I'll give you a moment to think about that, and feel free to chat to the person next to you as well. Okay, so the majority of the class are saying option D, so which I agree with. So so the idea here is that the orientation selectivity of cells in V1, the fact that simple cells will respond to an edge that is, some of them will respond to just a vertical edge rather than a horizontal edge, for example, comes from the pattern of excitatory and inhibitory signals that they're receiving. The presence of that inhibitory surround means that as the edge is rotated, it will begin stimulating the inhibitory part of the receptive field, reducing the response of the simple cell. And so if we don't have any inhibitory signals entering V1, presumably what will happen is that the cells there will lose their ability to detect the orientation of edges. But you would also expect there to be an elevated response to diffuse illumination, so there's just sort of broad light falling on the retina, then the lack of these inhibitory signals should lead overall to a greater sort of excitatory response in the simple cells. I've also talked about how the responses of cells at one stage of visual processing, like the simple cells, can be understood in terms of how they integrate signals from earlier stages of processing. And so correspondingly, if we're losing the ability of cells to detect edges of a particular orientation in V1, that may impact on the ability of later stages of visual processing to detect more complex patterns of information as well. And while there may be greater excitation of V1 cells because there's a lack of these inhibitory signals, that wouldn't lead to a improved detection of the orientation of edges for the reasons that we've just discussed. Okay, so let's have a quick break and come back at 5 minutes to 3. Thanks. Okay, so I better get started again. So we've been talking about the occipital cortex at the very back of our head, where we have this region of our cortex called area V1 that's receiving signals from our retina relayed via the thalamus. There are other parts of the occipital cortex as well that are involved in vision, and some of these are referred to as V2, V3, and so on. To highlight one or two of those, so in V1, we've just been talking about how there are these simple cells that pick up on the presence of edges in our visual field that have a particular orientation. In V2, we have neurons that are receiving signals from V1 and detecting slightly more complex patterns that are present, like these slightly more complex contours that are represented here. And then one other area that I'll highlight is called V5. It's also known as area MT, the middle temporal area. And this contains neurons that also receive input from V1, but are able to detect the presence of movement in our visual field. So anytime we see movement, like right now when you look at that moving object on the slide, there are neurons in area MT of your brain that are becoming active. Patient LM that we talked about at the beginning of the lecture suffered damage to her cortex, and it's generally thought to be damage to this specific area MT, that then led to her inability to see the movement of objects around her. And there's even evidence that electrically stimulating neurons in this area can cause someone to see motion differently. And so, to give you an example, what this study did is it studied motion perception in monkeys. They showed the monkeys, or they trained the monkey first of all to report what kind of motion it's seeing. So it would do a task where they're showing it sort of a pattern of dots that might be moving towards the right or moving downwards, for example, and they train the monkey to respond based off the direction that the dots are moving. They then recorded from individual cells in area MT using an electrode and found that there were cells there that responded based off the direction of motion that the monkey was seeing. So some cells would respond when the monkey is seeing the dots moving towards the right. Other cells would respond when the monkey is seeing dots moving downwards. And then in the final stage of the experiment, they got the monkey to do this task, but they then electrically stimulated particular cells. And they found that this seemed to affect the direction of motion that the monkey saw. So for example, in a trial where what's been shown on screen is a set of dots moving towards the right, they would electrically stimulate neurons that usually respond to downwards motion, and then the monkey responded as if it was seeing downwards motion rather than rightwards motion. So it's evidence of a kind of causal relationship between activity of neurons in this area and the actual direction of motion that is perceived. Area MT also becomes active when we experience illusions of motion, and this is one example. So this is something called the rotating snakes illusion. The image is not a GIF or a video, it's just a static image, but the design of the image seems to trick our visual system. So usually what people experience is that they have this kind of sense of like rotating motion, particularly if you kind of move your eyes across the image. And when we experience this kind of illusion of motion, area MT is becoming active as well, so suggesting that it has this key role in our perception of motion. Cats also appear to experience this illusion, so you can try this out with your cat at home if you have one. Which suggests that the cat's visual system is detecting motion in a similar way to how ours is. My cat Sunny was helping me prepare the lecture yesterday, and she was quite interested in this illusion. But she was also interested in pretty much anything else that came up on screen. So, so it shows the importance of a control condition. Okay, so area V1 at the back of our brain is receiving signals from our eyes and then forwarding those signals to other regions of our cortex. Outside of the occipital cortex, there are two main pathways that these signals take. So there are signals sent from V1 across the top of our head into the parietal cortex, which is referred to as the dorsal pathway of the visual system. And then we have another pathway where signals from V1 are sent into our temporal cortex, and this is called the ventral pathway. A really influential idea for understanding the organisation of our visual system and the function of these two pathways was proposed by Leslie Ungerleider and some of her colleagues. And her idea is that the ventral pathway of our visual system is primarily involved in the identification of objects, so allowing us to recognise what kind of object that we're looking at or who the person is that we're looking at, for example. Whereas the dorsal pathway is primarily involved in representing the location of objects around us. And so for that reason, these are sometimes referred to as the what pathway and the where pathway, respectively. Our sense of the location of objects is also really important for how we interact with the environment around us, like our ability to reach out and grasp an object or to navigate around obstacles when we're kind of walking down the street. And so sometimes the where pathway is also thought of as being like a how or kind of visually guided action pathway. To help illustrate this idea a bit further, one of the original experiments that Ungerleider and her colleagues ran was in monkeys, and she trained the monkeys to do two types of cognitive tasks or visual tasks. One was a landmark discrimination task, where the monkey would be presented with two boxes, one of which would contain food, and there'll be some other object like this cylinder located nearer to one of those boxes, and the location of that cylinder would predict which box contained the food. It would always be located nearer to the box that contains the food. And so once the monkey has experienced this and kind of learnt that rule, it becomes a very simple task as long as it's aware of the relative location of the different objects. In a second task, object discrimination task, they would, for example, present the monkey with an image, like of a triangle or a square, and then the monkey would have two different objects it could choose between, and if it chose the matching object, matching the image that was shown, then it would be rewarded with food. And so here, to do the task, it's very simple. Once you've kind of learnt how it works, all you need to do is be able to recognise the shape of objects. Then what they did is, once monkeys had been trained on both of these to perform really well on both of these tasks, they removed part of the cortex of the monkey's brain. In one group of monkeys, they removed a part of the parietal lobe, so disrupting the dorsal pathway of the visual system. And they found that this led to impairments in performing the landmark discrimination task. So the monkey no longer seemed able to pick up on the kind of relative location of these different objects. Whereas in a different group of monkeys, they removed part of the temporal lobe, disrupting the ventral visual pathway. And they found that these monkeys now struggled with the object discrimination task that required them to match the shape or be aware of the shape of objects. And importantly, there was a double dissociation, so meaning that monkeys that had part of their parietal lobe removed, even though they struggled with the landmark discrimination task, they could still do the object discrimination task. And monkeys with their temporal lobe removed could still do the landmark discrimination task. And so it suggests that these different aspects of our vision, our ability to recognize what we're looking at, like the shape of an object that we're looking at, and our ability to see the kind of location or be aware of the relative location of objects around us, appear to be served or produced by different areas of our visual system that are able to operate at least partly independently from each other. In humans, there are also many cases of people who have suffered damage to either the dorsal or ventral visual pathways of their brain, and this can occur following a stroke, for example. One quite peculiar symptom that can occur in people who have suffered damage to the parietal visual areas, the dorsal pathway, is something called spatial neglect, which is this apparent inability to pay attention to a particular part of our visual space. And that's described in this video.
Yeah, because Alan's middle often isn't it?
The middle is the right.
You must always eat on the right side, but not on the left side. Just leave that.
Give her a dinner plate, and she'd only eat the vegetables and leave the meat, which is disappointing because I was a cook.
So if he starts a picture, it can not fit in the paper if he gets over to the left, because he hasn't left enough room.
Spatial neglect is a syndrome, that is signs and symptoms that patients most frequently have after stroke, but sometimes in forms of Alzheimer's disease, where they ignore one side of space. So people, objects and sounds on one side tend to get ignored, and the patients who have it tend to be drawn to the other side. And this can extend to things like the food on the plate in front of them, that they'll only eat food on this side and then say they're finished. It may extend to parts of their own body, so when trying to shave, they may only shave this side of the face and miss out this side, put makeup on.
So you can only see from across there?
Yeah, from there to there. That's my vision.
So what's going on in the other half?
Nothing.
There's nothing at all?
Because often I've come up from this side to give you something.
Anybody
haven't
can
I?
creep up my left, I wouldn't
And
know
he
they were
doesn't
there.
see anything.
I can't sense them.
So if you just look at me and just point at the finger that moves, okay? Or say both of both. Tell me which finger moves.
First one. That one.
Okay.
Yeah.
Look at me.
That one.
Okay. Look at me.
That one. That one. That one. That one. That one.
Great. That's fine. They tend not to describe the condition, to be honest. One of the most intriguing things about it is that this sort of loss of attention for one side of space doesn't leave a great gaping hole, and that's why it's very useful to contrast it when someone's got what's called a hemianopia, which is a primary sensory deficit. They can't see that side of things, and they know they can't see it, so they're constantly moving their heads like that. Whereas patients with neglect are unaware of their deficit, that they're not even aware that they're missing something, because their sort of imagination space has gone on that side too.
Does he have a name?
I haven't thought of one.
I can stand right next to you, can't I? You can't sense I'm there.
No, you can't.
If you told her to look, she'd actually look around that way, like that, right round on the right-hand side, and then come back to central. She wouldn't actually think about coming and looking round to the left as well.
People often say, why don't you just tell them to look to the left, and relatives can be very frustrated about this too. And I think you've got to be aware that the process of attention and these higher visual areas are involved in the representation of space rather than just seeing things where they are. See the lines there?
Split them in the middle.
Yeah, that's one end, that's the other end. Show me where the middle is. Mark the middle for me.
I was only looking at a line from there to there. It's in the middle of my line, not the middle of your line.
So the first time I saw you, that's where you were marking the centre of the line as well. Patients realise something's wrong when things are pointed out to them over and over and over again. So in a way, I think for them it's slightly abstract, that they know that there's some problem on the left-hand side, and they're not aware of that problem, but they know everybody's telling them they have to do something more on the left. And so they use strategies and things like that to try and overcome the problem without really being able to appreciate it in its fullness.
I remember getting those word search books, and I remember it was only one side was always ever filled in. Yeah, and then slowly she got better and better at it, because I used to say to her, Colby, you know, I'm not getting value for money here, you're only doing half the page. Yeah, so slowly she learned that, do the left side first and then come back to the right.
Some people have it soon after their stroke, and it gets better within a few weeks. And then another group of people have it long term and learn strategies to overcome it. So if you probe deeply, they still have problems with attention, even that bias may go away, but they still have deep problems when trying to find things, and then they also tend not to remember where things are after they've looked at them. They have poor working memory for space as well. So a lot of these deficits don't recover, although people can use strategies to overcome them.
Give that the police if they find you. It's good.
Okay, so in that video, the man and the woman has this tendency to sort of ignore the left side of their visual field, and that tends to occur when there's been damage to the parietal cortex on the right side of the brain, whereas people who have suffered a stroke that's caused damage to the left side of their brain are more likely to ignore the right side of their visual field. Damage to the temporal cortex instead, so the ventral visual pathway, can cause quite different symptoms. And in particular, similar to what we saw in that study of monkeys, it leads to problems with recognizing objects. And this is, the clinical symptom is referred to as agnosia. And that's illustrated in this video.
In this short film, we look at the perceptual problem of agnosia. Agnosia is the loss of ability to recognize objects, people, sounds, shapes, or smells, even though the specific sense is normal and there is no memory loss. Here we see there are many objects around the sink. Greg has to be able to recognize each one and how it is used. For a person with agnosia, this task can be very difficult. Notice how Greg not only looks at the toothpaste, he also feels the tube in his hand. He then makes a mistake by reaching for the comb rather than the toothbrush. He knows the object is not right for the task. It takes a few seconds for him to work this out. He then chooses the toothbrush correctly. Next, Greg is looking for his deodorant. He has to look, smell and feel each item before he can correctly select the one he needs. This task could have been made easier for him by only placing the items he needs beside the sink. Too many items can be confusing or distracting.
Okay, so you can see here that he's not blind. He's able to see the objects well enough that he can actually reach out and grasp them correctly, right? But somehow, despite being able to see the objects, he's not able to easily recognize using sight alone whether this object is a toothbrush or a comb, for example. And this is another illustration of agnosia.
Evan suffered brain injury after being hit by a car a few years back, and that caused him to lose the capacity to recognize everyday objects. We asked Kevin what he sees when he looks at things.
I see colours, shapes, punctuated by faces, the faces of people which I really recognise, and they stand out much more than anything else. That's why I can't read words or read music anymore. So I still can imagine what it looks like in my mind's eye, but I can't imagine what it looks like, or I can't see what it looks like when I look at it. So essentially, I find things through colour.
In fact, the brain needs both the structural and semantic processing to be able to interpret information transmitted through vision.
I can sometimes pick out small components of an object, and by using memory as a reference, infer what it might be. Like if something's long and silver, it could be a spoon or a fork or a knife, and usually I'll use my hands to touch the end of it, so I know, oh, it's a fork, or it's a knife, or it's a spoon. So memory does play an important role in that, and in everything I look. I mean, I come to this chair, I know it's a chair, I don't know what type of chair it is, I know it's pink, but I was able to sit down in it. But this is my house, so I know where everything is. That's my son, my oldest son, was Santa Claus. Santa Claus. And this is one of my youngest son. When he was younger, he's a teenager now. Yeah, that behind that angelic face is a demon child. That's my eldest son when he was, must have been about 6 weeks old, 2 months old. And I think that's me. And that's Aiden, my youngest son, when he was a baby.
And do you see the details of the face? Like, do you see the eyes clearly? What is it in the image that you recognize?
The whole face. And I'll tell you why you can see. I can see a whole face, but I can't see a whole object. Like this thing here, I only see parts of it. Like, I see there's diagonal things here, but I can't see the whole thing. But with the face, I see the whole thing. So you say, well, how come I can't see that in its entirety, but yet I can see the face in its entirety? It's because a completely different center of the brain processes faces and processes objects. And in me, the part that processes objects, symbols, has been damaged. But the part that processes faces is fine. And that's how I'm able to recognize people's faces. So I recognize my colleagues at work, my family. That's much, I'd much rather have the condition I have. I'm lucky, because I'd rather have that than not be able to recognize faces. That would be awful. Not being able to see the people you love anymore.
Visual.
Okay, so again, he's clearly not blind. Like he can recognize people, for example. And when describing his inability to recognize a fork, he said he can see the color and like aspects of the shape of the object. And it's not a problem with memory. He says that like when he has the chance to kind of feel the fork, he can infer that this must be a fork rather than some other kind of object. So it's not that he's just forgetting the words for what he's looking at, but rather there's this stage of vision after our brain has kind of extracted the elementary features of an object, like the edges that are present, the colors that are present, and so on, where that object is then those features are then kind of grouped together into a kind of meaningful whole. And that seems to rely on neurons in our temporal lobe. And then at the end of the video, he mentioned this other condition where people have difficulty specifically with recognizing faces rather than other types of objects. And this is a type of agnosia called prosopagnosia, and it's illustrated in this video.
Not even a clue who that would be.
Penelope Cruz.
Oh, Oh, really? So if I saw Penelope Cruz, I would look for the nose. Okay. And because that's head on, you can't tell. I know she has a big nose.
I'm sure she'd be pretty pissed you just said that.
I know she would be, but sorry, it's true.
So if you saw your wife's face cut out of a magazine, you would know it was hers?
Maybe. Maybe. Perhaps if it was out of context, I might have a problem with it.
If I was to leave and to come back with another woman, do you think you could distinguish my face from hers?
Absolutely.
Let's test it out.
Bring it on. It's tougher than I thought. And I'm going to say Hillary is the one on my left. I would say that is Hillary. Mostly I think it's Hillary because of the smile. And I think I remember the smile.
You're wrong, Jeff.
I can't believe it.
So sorry.
I can't believe it.
Are you a normal guy?
I would say I'm a normal guy.
So this is just something you've learned to cope with?
It's something I've pretty well learned to laugh off.
For somebody who can't recognize faces, you've definitely chosen a very interesting profession.
True, in retrospect, yes.
You're a photographer.
I'm a photographer, photojournalist. I've been for years, and I'm a photo editor of the National Post.
Okay, so I mean, the experience of like finding it difficult to recognize people is something that probably some of you can relate to. It's something that just varies between people in general. In people with prosopagnosia, it can also vary in severity as well, but for some people it can be very severe, that they have difficulty recognizing, close family members that they see every day, and even sometimes don't recognize themselves in the mirror, for example. When we zoom in on the ventral pathway of the visual system, what we find is that there are different kind of sub-areas that we can refer to as modules that seem to be responsible for processing different types of objects that we can recognize. And so one of these that has been studied in a lot of detail is called the fusiform face area, or FFA, which is located along this kind of ventral surface of our brain. And as the name suggests, there are neurons here that seem to be specialized to detect faces and recognize faces of people that we have seen before. This area, the FFA, has been studied a lot in humans. There's also a lot of work looking at cells in the visual system involved in detecting faces in monkeys as well. And in monkeys, there's a region of the brain called the inferiotemporal cortex, or area IT, that is also in the temporal lobe, and where there are also cells that seem to be specialized to detect faces. To give you an example of that, what this is illustrating is data from a study where they were recording from individual cells in area IT of monkeys, macaque monkeys, and showing the monkeys different images. And they found that these cells would produce lots of action potentials whenever the monkey was looking at some type of face, like photographs of human faces or other monkey faces, but then didn't tend to respond when the monkey was looking at other types of complex visual objects or scenes, like a picture of like some flowers or a house and things like that. This is another example of cells that would respond whenever the monkey saw different images of faces, but then didn't tend to respond when the monkey saw different images of other kind of categories of objects, like bodies where the face isn't visible, sort of natural objects like fruits, or sort of artificial objects like gadgets, and so on. And this is one more study sort of illustrating the behavior of these types of cells in the visual system, where what this is representing is different types of images that they would show to the monkey during the study. And in blue here is a representation of how much a particular cell they were recording from was responding, producing action potentials. And here what it's illustrating is that this cell will respond when the monkey sees some type of face, like even a sort of cartoonish looking drawing of a monkey. but will also respond when the monkey sees this kind of schematic or symbolic representation of a face, something that captures those core features of like 2 eyes and a mouth, even though it's not really a face. But then the cell stops responding when the monkey sees very similar images, but where some of those core features are removed. And so what it suggests is that this cell is responding to the kind of overall holistic pattern of a face, like the presence of 2 eyes and a mouth that defines what a face is. And we saw how earlier in our visual system there are neurons that detect simple visual features like dots of light or edges in particular locations. And so it may be that this type of neuron is integrating together the responses of those neurons detecting sort of like a horizontal line in one part of the visual field, for example, in order to detect this kind of overall pattern that faces often have. Some of the work that has been done in understanding how the brain is processing faces is from the lab of Doris Tsao, who's at UC Berkeley, and I've linked a TED talk here where she talks about some of her work. I'll just show a short excerpt of it here. And so what you'll see in the video are, again, just images on a screen. I'll play a bit of it to start with. And this is what a monkey is seeing during the experiment, and the crackling sound you hear in the background is a representation of when a particular cell that they're recording from in area IT is producing action potentials. So what you should notice there is that this cell is responding to something really specific. It's responding to this one particular person, which is this blonde guy. And it responds whenever there's a photo of that guy, even if he's pictured from different angles. And it doesn't respond to photos of other objects like a clock, and it doesn't respond even to photos of other people. And so this cell seems to be detecting the presence of a very specific person and presumably plays some kind of role in the monkey's ability to recognize people or other monkeys based off their face. So the FFA in humans, which also contains cells that respond to faces, it becomes active, this region of our brain, whenever we're looking at other people and seeing their face. It also becomes active when we see other types of faces, if you like. And so one example is face pareidolia, this experience of seeing everyday objects that look like faces. And so there are many, many examples of this. There are, groups on like Twitter and Reddit and so on that have collected many hundreds of photos like this of everyday objects that people have come across that just look like a face. And it's interesting from a psychology perspective as an example of how our brain can kind of impose a certain type of social meaning on the world around us. And so the FFA becomes active when we see these kind of illusory faces, if you like, in objects around us. It also becomes active when we see other kinds of images that represent a face. So these black and white patterns are called Mooney faces. You can probably see that there's a face present in each of these images, even though the images are not really like sort of photos at all, like regular photos. And this area, the FFA, becomes active even when we just kind of imagine a face, like we picture our mum's face in our mind, for example. And so activity in this area seems to be closely related to our perception and ability to detect faces around us. There are also other modules, other areas in the ventral pathway of the visual system that seem to be specialized for processing different types of objects. One example is something called the extrastriate body area, or EBA, which is a region that contains neurons that become active when we see parts of a body, like somebody's arm, for example, or the kind of overall shape of the body, regardless of whether the face is visible or not. But doesn't respond as strongly to faces when the body isn't as visible, or other types of objects. And then another example is something called the parahippocampal place area, or PPA, which becomes active when we see places, so meaning pictures of like landscapes, particular buildings, particular locations that we recognize within a building, and so on, and responds to these kind of places rather than to specific objects. So to sum up what we've talked about, we've seen how information from our retina is sent to our cortex initially to a region at the very back of our brain, the primary visual cortex, where there are these simple cells that pick up on very basic patterns that are present in that light falling on the retina, like the presence of edges in different locations of the visual field, and detects the orientation of those edges. And then that basic information is then forwarded on to different parts of our occipital cortex and along these two main pathways towards our parietal lobe and towards into our temporal lobe, the what and the where pathways. And in the temporal lobe, we find these different modules, different groups of neurons that are specialized to process information about particular types of objects, like faces. So the key points here are that signals from V1 are sent partly along a dorsal stream of the cortex, which seems to be specialized to process the location of objects, and a ventral stream, which seems to be specialized to identify objects. And in general, as we travel further away from the retina, deeper into our cortex, we find cells that are responding to more and more complex visual information. Okay, so to finish up, we can do one more poll everywhere question. So the question here is, when chatting to your friend over lunch, which parts of the visual system are likely to be active? The primary visual cortex, the ventral visual pathway, the dorsal visual pathway, tracks that are running between the thalamus and the cortex, or all of the above. Okay, so it looks like everyone has put all of the above, which, sorry, yeah. Sure, yeah, I'll just be a moment, yeah. Which, yeah, I agree with as well. So the idea here is that our visual system has this hierarchical structure. When we're chatting to our friend over lunch, there'll be early stages of the visual system picking up on very basic features of what we're looking at, the presence of edges along their face, for example. Our ventral visual pathway will be active to identify what we're looking at, recognize their face. Our dorsal visual pathway will be representing information about the location of objects around us. And there's also this information traveling between the thalamus and the cortex. Okay, so if you have any questions, maybe come up and ask me. Otherwise, I'll see you in a couple of weeks' time. And so, yeah, so I'll see you in week 7 for the midterm test. Thank you.